So here we are to discuss on Excel biochemistry section, and in this program we'll have several strategies. So this is ACPL strategy. That means always, chances, try only if you can, and leave. If you join our program, we would be letting you know about all the topics which are very essential for you, which have certain chances of coming, and least ones can be seen to try only if you can, and whichever are not so frequently asked in the questions would be tagged at the lead. But today we would be talking about cell. So I would like to ask you guys, what is cell? What exactly would you mean by cell? So cell is basically the structure. We all have heard about this structural and functional unit of life. So what exactly makes it so? We all know that there are multiple unicellular and multicellular organisms. So basically, the cell itself, in the very beginning, is capable of initiating life on its own. Like you talk about organelles, you talk about cytoplasm alone. These in these individualities, all these structures won't be able to sustain life, whereas a whole cell can. So we call it the structural unit of life. And why the functional? Because uh, it is called a functional because a single cell itself, you see, is a unicellular organism. A single cell itself is able to carry out all the living functions, be it respiration, be it reproduction, anything and all is carried by the cell itself. Therefore, it is the structural and functional unit of life. It comes in various shapes, sizes, specialized functions, but all in all, it is the cell itself that will give the meaning to life. So, who discovered cell? Anybody?
Robert Hooke.
Very well. So, yeah, the cell was discovered by Robert Hooke, and nucleus was observed by nucleus was observed by Robert Brown. Moving forward, we will talk about cell theory. So now, what is cell theory? Who gave it? Schleiden and Shawn in 1839 gave the cell theory, whereas it was later modified by Rudolf Virchow. Now, what exactly the cell theory speaks of? It says that the all the organisms are made up of cell, and a cell will only give rise. A pre-existing cell will only give rise to the new cells. Cellular is cellular, unicellular is cellular. Carrying forward, we are going to talk about the types of cells now. So there are two types of cell. One is prokaryotic, the other is eukaryotic. What is the basic difference between these two? It is the packaging of the genetic material. In prokaryotic cells, the genetic material here is not enclosed in any kind of membrane. It is lying open in the cytoplasm and hence is referred to as nucleoid. Whereas at the same time, in eukaryotic cell, the genetic material is well packaged inside a double membrane organelle known as nucleus. So this is the basic difference between the two types of cell. Along with this, we talk about that the prokaryotic cell, which organisms can be referred to as prokaryotic, the bacteria, mycoplasma, cyanobacteria, that is blue-green algae, all these are termed under prokaryotic cell, whereas the eukaryotic cell form the higher organisms like protists, plants, animals, fungi, these. So there are several kind of differences between these two. The prokaryotic cell is quite smaller in size as when compared to the eukaryotes, and the cell division carried out in prokaryotic is through binary fission, mitosis, and budding. Uh, and whereas in eukaryotic cell, it is carried out by mitosis and meiosis, both undergoing a proper cell cycle and all these steps into it. Next, moving forward, the genome or the genetic material, how is it? So the genetic material present in these prokaryotic cells, so the genetic material present in these prokaryotic cells is is the DNA is with non-histone proteins. So now what are histone proteins? Histone proteins are positively charged proteins around which a DNA is wrapped. But this is lacking in this prokaryotic cell. It is not present in the prokaryote. These proteins are not present in the prokaryotic cell. Hence, this is a very major difference, one of the major differences between these two. So, in the prokaryotic cell, the DNA is not packed with histone proteins. And the other one that I told you, that this material is not enclosed within any of the membrane. Whereas in the eukaryotic cell, the DNA is moves around several histone proteins and it is packaged inside a double membrane nucleus. Um, the nutrition mainly here is through absorption or maybe photosynthesis, whereas in eukaryotic cell, it could be either through uh, absorption, ingestion, photosynthesis, plants, example in plants. There, so the cell wall also differs in these two cells majorly. The cell wall present in the prokaryotic cell is made up of lipopolysaccharides, mostly or mucopolysaccharides, whereas in eukaryotic cells it is made up of chitin, cellulose, in plant cells and fungi especially, whereas the animal cells lack the cell wall. Now, moving on to cell organelles. Now, what are cell organelles? Cell organelles are nothing but the living parts of the cell. We will discuss a few of these. First would be nucleus, very important. So, uh, in nucleus, here we see it is a double membraned enclosed organelle. So, it has a nuclear envelope, it has pores also, and inside of a nucleus is a nucleolus as well. In this is a cavity present, and in this cavity around the nucleolus are present the chromosomes, which is the actual genetic material with, packed inside the cells. Ask me anytime if you guys have doubt or if you would like me to elaborate on any of the points, please. So yes, this nuclear envelope, that is, it is basically around 70 to 80 angstrom in thickness. There is a space between the nucleolus and this outer membrane, which would be about 200 to 300 of angstroms. This space here around, inside this membrane is called perinuclear space. Yes, it's called perinuclear space. So basically, here what happens in nucleus is nucleus carries your genetic material. At the time of cell division, during the cell division, this nuclear membrane dissolves inside the cytoplasm, where and the whole cell would only be covered by the cell membrane. This envelope dissolves, and these chromosomes are not present as chromosomes. They are present as fine threads of DNA, which are referred to as chromatin. So these chromatin, when a cell undergoes the cell cycle, they condense in a series of steps and then they form chromosomes, which further take part in the cell division. So nucleus is very important. Nucleus plays a very important role in cell. Moving forward, ribosomes, it is another differentiating factor between prokaryotic and eukaryotic cell. In a prokaryotic cell, the ribosomes which are present are of 70 S subunit, whereas in eukaryotes, it is of 80 S subunit. So here it can be divided into two subunits. This is a large subunit and this is a smaller subunit. Basic function of ribosomes is to make a protein out of this mRNA. How is this mRNA generated? That is another whole different process of translation and transcription. So this transcription occurs when the genetic material that we talked about here gets transcripted into an mRNA. Then this mRNA binds with a ribosome. This undergoes a series of actions which we would call as uh, translation, and is converts this mRNA into functional proteins. So this is the basic function of ribosomes here. And it has different sedimentation rates, which according to which the size of it is determined. Moving on, we have chloroplast. So this chloroplast is basically present only in the plant cells, the cells that can actually undergo photosynthesis is due to this structure called chloroplast. Again, this is another double membrane structure. So we have three double membrane organelles in our cell. That would be nucleus, chloroplast, and mitochondria. So chloroplast being one of them. It is also double membrane structure. It has an inner membrane and an outer membrane. Inside this, there is hollow space, which is filled with a matrix called stroma. This contains disc-like structures. These disc-like structures are called thylakoids, and the whole pile of this structure, like all these pile of these discs, is known as collectively known as granum. Now, these granum are connected with one another through lamellae. So this is the basic structure of chloroplast, and the photosynthesis occurs in two phases. That is, one is light dependent and light independent phases. Uh, Chloroplast takes care of all those. We would have our mitochondria. So mitochondria is also known as the powerhouse of the cells. So here, this basically is responsible for the respiration, oxidative respiration that takes place inside a cell. It is also another double membrane structure, the outer membrane and the inner membrane. The inner membrane is folded into these finger-like projections. These finger-like projections are known as cristae, and the inside of the cristae is filled with a matrix, and hence this is the structure of the mitochondria. Uh, the number of mitochondria per cell is about 42 to 100 something per cell, and it takes part in oxidative respiration. This mitochondria carries out a series. It is very important as it generates the energy that is required to make the cell viable. Without the energy produced by it, so a cell won't be able to sustain, and hence the whole organism won't be able to sustain. So this plays a very important and crucial role. So basically, if you see any of the last year papers, the questions basically come from the organelle sections, like which could be, it could also come in match the following types, which organelle would be double membrane or single membrane, what is the function of it, and if any of these cell structure, like basic differences in prokaryotes or eukaryotes, it could come in multiple select type questions. So these are very important and critical questions that come from this section.